Genome sequence is the complete list of the nucleotides that make up all of the chromosomes of an individual or a species. Within a species, the vast majority of nucleotides are identical between individuals, but sequencing multiple individuals is necessary to understand genetic diversity. And as techniques for mapping have gotten more complex and more complete, we've gotten quite a great deal of information. So if we look at bacteria, for example, versus archaea versus eukaryotes, eukaryotes tend to have a much larger genome, but the number of genes don't seem to be roughly a parallel within that structure. Okay, eukaryotes tend to have a lot lower gene density within this structure, the genes per megabases. So overall, genomes are larger in eukarya, but the number of genes, while larger in eukarya because of such a massive difference, the density is not that high. Okay, so eukaryotes have a lower gene density. Introns and exons, you might remember those, introns and exons, we see that in eukarya, and that's going to be a big indicator of what's going on there. Archaeans have some eukaryotic traits. They're sort of odd. So you can see here that introns are present in some genes. And non-coding DNA, bacteria and archaea don't have much of this, but eukarya has a great deal of repetitive non-coding DNA. Acquisition of the full genome sequence, we estimated that there are between 50,000 and 150,000 genes within the human genome. We know now that the closer answer now is about 46,000 with a few thousand microRNAs as well. Overall, though, only a very small fraction of the genome, about 1.5%, are exons, the actual protein coding sequences. The rest of it is associated with non-coding RNA genes, regulatory DNAs, and lots of other structures. About 44% of our non-coding DNA segments catalyze their own replication and movement throughout the genome. So we call these transposable elements. Transposable elements move around. They are the jumping genes, and they have their own slide in a second. Within that, you have lines and signs. Lines are the most common, the L1s are the most common lines in humans. These are long interspersed nuclear elements. These are going to be duplications of long stretches of base pairs. The Y chromosome, for example, is mostly made up of those. Then we have the short interspersed nuclear elements. These things like ALU elements are also going to be part of that process. Overall, these repeats seem to contribute to genomic instability and evolution, and so they are controlled through methylation and histone modification and pRNA, which we'll talk about in a minute, all of these things, but they move around. They are transposable. We have another 14% of our genome that is non-transposable. They do not catalyze their own process and reaction. These are going to be repetitive DNA sequences, and you're going to have short ones and long ones. The simple tandem repeats are an example of the simple sequence DNA. Those are the ones you look at when you do electrophoresis and gene mapping and DNA fingerprinting. And then the large segment duplications are going to be sort of stretches of duplications that repeat but don't move. Okay, outside of that, you also have things, these unique non-coding DNAs, which typically are pseudogenes and gene fragments. Pseudogenes are DNA sequences that are related to known genes that have lost their protein coding ability, whereas gene fragments are one that have mutated and are damaged or have pieces of things, but we may not actually know what gene structure they're connected to. And then you have the regulatory sequences and the introns that are going to be part of that. That's going to include things like promoters and enhancers and all sorts of structures. Okay, there's even some portions of these things that are old viruses that became part of our DNA and all sorts of structure. So we see that most of our DNA is linked to repetitive structures, but very little of it actually has to do with making a protein. Prokaryotes and eukaryotes have stretches of their DNA that can move from one location to another within the genome. These things are called transposable genetic elements or transposons, jumping genes. The first evidence for these came from American geneticist Barbara McClintock's breeding experiments with Indian corn in the 40s and the 50s. And as she tracked these plants through multiple generations, she found color changes that could be explained only by the existence of these moving genes. This was met with skepticism It wasn't until 1983 that she received the Nobel Prize for her work because it was validated. Eukaryotic transposable elements are of two types. You have transposons, which move within a genome by sort of a cut and paste process. It removes the element from the original site, or it might even copy and paste. They can happen. The mechanisms require enzymes to do this, and it's called transposase, which is encoded by the transposon. But we also find that there are another type, the retrotransposons. These move by an RNA intermediate. Retrotransposons always leave a copy at the original site because they are transcribed into RNA. Then a special enzyme is used, reverse transcriptase, which turns that back to DNA, which then can be then inserted into a new location. That enzyme becomes incredibly important when we get to biotechnology. So reverse transcriptase reverses the DNA RNA protein process, taking RNA back to DNA. L1 sequences and ALU sequences, these are ones that use these structures, but they are defined by this capability to change their location, and that is something that is really key to evolutionary change. Gene family is related to these ideas. It's a set of several similar genes that have formed from duplications and mutations and changes that have built up over time. One such family of genes for this are human hemoglobin. There are 10 genes in 2 clusters on different chromosomes for hemoglobin. We call these the alpha and the beta globin families. These 2 clusters are thought to have arrived through duplication events about 500 million years ago, and they are clustered into families based on shared sequences. This relationship, these multi-gene families, become really important for genes that shift over time, especially developmental changes. If you look at the example here, the type of hemoglobin that's being produced by the embryo is different from the type of hemoglobin that's being produced by the fetus or by the adult. That makes sense because the way that you interact with oxygen is very different in those different situations. Another very common gene family is the ones that make up the rRNA genes. They're all in one transcriptional unit. So whenever you need to make a new ribosome, you transcribe all of the parts, which are then assembled at the same time. You don't make them in 3 different parts. That doesn't make sense. Okay, so this has evolved over time. It's an efficiency relationship, which is why it's supported and enforced by natural selection. You actually start looking at regulation, which is the focus of the rest of this section, the first major type of regulation is one that we've been talking about since the energy unit, which was a while ago, and that's metabolic regulation. Both prokaryotes and eukaryotes can alter their patterns of gene expression in response to changes in environmental conditions. Okay, so you can alter what you're producing based upon the precursors and so on. So feedback loops and regulation, all of these things are going to play a role into what's going on. If you go beyond metabolic pathway regulation, then it gets significantly more complicated. And now let's look at what that does to a bacteria. We need to remember that transcription and translation occur at the same time in bacteria. So what we're looking at here is groups of genes that control and regulate genes in bacteria, and we call these operons. Okay, so the operon model, we've been knowing about the operons since the 60s. These are groups of genes that control and regulate gene regulation in bacteria. Eukaryotes have operon-like structures because we have groups of genes that work together, but we don't have operons because we cannot transcribe and translate at the same time. They happen in 2 different places. But when you're looking at this, the operons can be classified into type of regulation and direction, basically. So negative gene regulation is going to be starting and stopping things, whereas positive gene regulation, which we'll look at in a little bit, is going to be slowing down and speeding up. It's not stopping, it's just going faster. Those negative gene regulations can be repressible, which means that whatever it makes is normally on and can be turned off, or it is inducible, so whatever it makes is normally off and can be turned on. The repressible operons tend to be biosynthetic. They're going to be making things. So what we see overall within this gene structure, I use the laser pointer, the regulatory genes are always going to be upstream of the operon. Why? Because you're going to make the regulator before you get to the gene. It makes sense. The slashes is just a distance here. So what we're looking at here is the tryptophan operon, the TRP operon. It makes the amino acid tryptophan. Our promoter is going to be where the enzymes, the RNA polymerase, bind. The genes are going to be making products, typically mRNA. So the regulatory gene, in this case, creates a repressor. This is a repressible operon. So that means that it's normally on and can be turned off. So in this case, the repressor is always produced, but it is not produced in an active form. The RNA polymerase will bind to the promoter. Okay, the promoter is going to be the binding site. The operator is where the repressor binds. Since the repressor is off, the promoter structure is not going to have a barrier. The operon is the binding in all of the genes. So since there is no repressor to stop this process, RNA polymerase will move forward and make all of the components necessary to produce tryptophan. So these are all the 5 enzymes it takes to make tryptophan. Okay, when tryptophan is present, it will bind to the repressor, turning it on, making it active. That can then bind to the operator, which acts as a physical barrier, stopping the polymerase from moving forward. You don't make tryptophan when you have an abundance. This is an efficiency thing, which is why natural selection is favoring it. Operons tend to be digestive. They tend to break things down rather than building things up. So we have here, this is lactose. This is the lactose sugar. When a bacteria takes in lactose, it's going to need to break it down. So you have the LAC1 gene, that's the regulatory gene. The green is a promoter. The regulatory gene is always made first, but this is an inducible operon, so the repressor is produced in an on form. It's going to bind to the operator and prevent the binding to the promoter for the actual product creation. Okay, when allolactose, which is the type of lactose that this is digesting, when that is present, that will bind to the repressor, turning it off. It is inducible. Okay, so we have to induce the repressor to be turned off. It will then no longer be able to three-dimensionally hold on to the operon. Polymerase will bind and produce the 3 segments needed to produce the enzymes to digest lactose. The last lactose that will be digested is the one attached to the repressor, which will then turn it back on and stop this process. So both of these are negative gene regulations because there is a start and a stop. But is it repressible or is it inducible? That's the question. They don't have to give you the same 2 operons. You need to be able to interpret them. Just realize that you're always going to go from left to right. You're going to follow the process. Regulatory genes come first. Promoters are where the enzymes bind. The promoters will also have operators, and that's where the repressors bind. The operon is the promoter and all of the genes necessary to do the job. Regulation is positive. This occurs when a protein molecule interacts directly with the genome to speed up transcription. The lac operon is a great example of this. So basically, if a bacteria has lactose and glucose, they're more likely to digest glucose first. However, if glucose is missing, okay, if glucose is scarce, then they're going to go to their second choice sugar, the lactose. It's more complicated, it's more energetically expensive to break down. So if it's present, it will be digested. But if glucose is not present and lactose is, you are going to have molecules that will bind and speed up this production. So you have the CRP. Okay, so CAMP is the secondary messenger that's used in glucose digestion. So if CAMP is abundant because it's not being used, it will bind to this CRP. That CRP will then bind to the promoter and upregulate the binding of RNA polymerase. When lactose is present but glucose is not, the lac operon will just do its normal job. So it's not that it stops, is it faster or is it slower? Okay, the CRP works on several operons that are encoded in enzymes that are catabolic, so it's not just with lactose, but this is a great example. Again, eukaryotes do not have operons, okay, but they do have many different ways of regulating their process. And why are there so many? Because we are separated into separate components. There are more points, there are more places that can be changed. So overall, we have a lower genetic density because we have differential gene expression. We can produce different things from the same base material. A typical human cell probably expresses about 20% of its genes at any given time. Highly specialized cells like nerves and muscles, even less. The differences are due to these regulatory control mechanisms. So where do they regulate? They regulate at any point throughout this process. So there are many, many, many different points of regulation that can happen. So with our greater complexity, eukaryotes have opportunities for controlling gene expression that a bacteria just does not have. Now, moving on. If we simplify this, there are really 6 major stages. So this is the simpler version. So keep this picture in mind. 3 of them are happening in the nucleus. 3 of them are happening in the cytoplasm. The first one is just straight up access. Okay, we can condense our chromosomes. We can make them tighter or closer. Okay, the more open, the more the gene can be accessed. So you might modify the DNA with methyl groups. You might alter the histones, like in this picture with acetyl groups. All of these things will prevent access to the genes themselves. So the first level of control we have is chromatin modification. This is going to play a major connection into epigenetics, and it's something that we're still trying to figure out how it really functions here. But it's all about altering the charges of the DNA and the nucleosomes, which is the bundle of DNA and histones together, or preventing access in the first place. Looking at transcription factors. Transcription factors are control elements that will alter what we are producing. They connect to control elements. So here we have the enhancers, which are the distal or distant control elements upstream, and then you have the proximal ones, which are right there next to the promoter. Okay, the control elements are binding sites for transcription factors. Only a few general transcription factors are sort of independently connected within this structure, and it's going to be leading to the gene product. Those control elements, those things that bind, they are going to lead to differential gene expression. So you're going to produce your primary RNA transcripts, your exons and your introns. All of these things are going to be controlled through these control elements, as we'll look at on the next slide. This is also going to be related to what we talked about with things like um, transposons. Transposons are connected to this because the transposons are being controlled by RNA, Pee-wee RNA, or you might be looking at introns and exons, which are controlled by small nuclear RNA. So these control elements are working with these RNA elements to produce different gene products, these different primary transcripts. Going to happen is that molecules are going to bind to the enhancers. These things are called activators. Okay, the transcription factors are going to come in. You're going to have these specific transcription factors that are for very particular jobs, and then you're going to have these general transcription factors that are going to be for all transcriptions, but they are going to bind to these control elements, and they are going to lead to an upregulation. Okay, the distal ones fold and become in line with the proximal ones, creating the perfect binding site for RNA polymerase. Okay, so the RNA synthesis will then proceed from the transcription factors down the gene. For us, we would typically look at RNA polymerase 2 here because that's going to lead to mRNA control mechanisms. Overall, the number of different nucleotide sequences found in control elements is surprisingly small. There's only about a dozen of them. And they work in combination. So here, for example, we have the DNA that's in both cells. Okay, the activators aren't shown as we can see, and they have different enhancers. Those enhancers are going to bind different transcription factors. Those different transcription factors are going to lead to the production of something very, very different. So in this case, we can make the crystalline, which is in the eye, or the albumin, which is in the liver, from the same DNA segment by altering the activators. So a particular combination of control elements is able to activate transcription only when the appropriate proteins are there, and they allow for regulation and control. In prokaryotes, coordinatingly, these genes is clustered into those operons. For us, these transcription factors are part of that process. Previously, that's also going to connect to the small nuclear RNA that we talked about before. The small nuclear RNA will come together to form the spliceosomes. The spliceosomes will produce the connection between the exons and removing the introns. You can change what is intron and what is exon, so some things will be produced, some things won't. The UTRs, the untranslated regions, are control segments as well, with that brown polyadenylation signal telling it how many poly A's to put at the end, and the 5' GTP cap at the top. Alternative RNA splicing significantly expands our gene repertoire. Between 75% and maybe even 100% of human genes have multiple exons, probably undergoing this process. Now, in addition to that, not only can you produce different introns and exons, you can produce completely different processes because of this. So the RNA splicing can be put together in different patterns to produce different things. Notice that they are still in numeric order. The only way that you can actually shuffle those things around would be to have a transposon, some sort of shuffling process. RNAs that will then interfere with gene expression. So if you get to the point where mRNA exists, okay, you really can either let it go on to make a protein or not. So non-coding RNAs can be used to interfere with RNA usage and reading, so we call it RNA interference. There are two major types of RNA that are involved in this process, miRNA, which is micro, and siRNA, which is short interfering. So these are small RNAs that can remodel chromatin, silence transcription, but also prevent the ribosome from reading or even destroy the target mRNA if it's something that's produced wrong. So during RNA interference, long double-stranded RNA is cut and diced into fragments called dicers, or the enzyme's called dicer that's cutting it into fragments. These fragments, these short interfering RNAs, in this case, you can see the short interfering being produced by dicer on the right, or the miRNAs being produced at the bottom. These will bind to the mRNAs. Okay. Both miRNA and siRNA can associate with the same proteins with similar results. The distinction comes to what they are formed from. So each miRNA forms from a single hairpin. You can see that picture here. Okay, while multiple siRNAs form from a longer structure. Okay, in addition, siRNA it pretty much only has one result. It's going to cut RNA that is incorrectly coded or active when it shouldn't be, whereas the miRNA has two options. It can either destroy the miRNA if the match is exact, or if the match is just close, it can block it. And that can then leave time for proofreading or verification or fixes. So a survey of species overall suggests that short interfering RNAs evolved first, followed by miRNAs, and later by the piRNAs that regulate transposons. And while there are hundreds of types of mRNA, there are thousands of types of piRNA. So this process is getting incredibly complicated as we look at more quote unquote advanced organisms. Point of control is at translation. So translation presents an opportunity for regulation. Okay, just like we had transcription factors, we have translation factors. Those translation factors are going to bind to the ribosomal subunits and encourage the mRNA's attachment. So there's a variety of these things allowing this process. Okay, translation of all of the mRNAs may be regulated simultaneously by the activation or inactivation of these processes. Okay, like some plants, for example, store mRNAs during periods of darkness and only activate them when light triggers these translation factors. Okay, so translation factors allow the proteins to be produced. If they're not present, the protein won't be produced, and we'll go on from there. The last major way to control it is to look at the protein itself, and they can either aid the protein or destroy it. Okay, a chaperonin is a great example of a structure that exists to aid protein processing and allow it to fold and form properly. However, if you have misfolded proteins, like what's going on the top here, this is an amyloid plaque, so it's a misfolded protein. This is going to be something that leads to Alzheimer's, for example. Remember ubiquitin. Ubiquitin can mark a damaged or misfolded protein, and that can either be taken by a motor protein to a lysosome to be digested, or taken up directly by a proteasome to be digested. So if you have a well-functioning protein, you can encourage that. If you have a misfunctioning protein, you can destroy it. Okay, so all of these conditions, all of these places have points of control, and they are all necessary to keep you functioning at your best. And when they fail, that's when you have mutation and disease and problems. There are many different points of control mechanisms that we can have here. Chromatin modification is all about access to the genes. Are we modifying the histones? Are we modifying the DNA? What are we doing to prevent or encourage access? RNA processing, that means that you're going to use your small nuclear RNA and your spliceosomes to alter and change the product. Okay, and then in addition, you could combine that with transposons to also shuffle those around, which is possible. Okay, if your RNA transcript is produced successfully, your next stop would be to integrate and control the mRNA itself. Okay, are you going to interfere with the RNA? That's fine. If you're using other RNA to do it, that's RNAi or RNA interference. Okay, translation control mechanisms are all about initiation factors, producing that's going to combine between these products. Transcription factors, that's going to be using those enhancers. Okay, those enhancers control elements, creating binding sites. Protein process, degradation control. There's all sorts of things here, and they're not in order, and I've just said them wrong, but you get the general idea. Because there are so many stages to our development, there are so many levels of control. For a bacteria, it's significantly simpler.